Okay, we are recording, just double checking. Okay, so the goal for today is I'm not going to be able to answer every single question that you might have. So I will refer you to Maria's office hours if I can't answer that question. We're trying to cover 9 hours of material today, so there's 9 lectures, and we only have 3 hours. So I'm going to be skipping some stuff if I feel like it was easy enough for students to understand, and I'll try to focus on the things that I believe are the hardest. So here we go. Lecture 21, Introduction to Metabolism, Biological Oxidation, Reduction, Reactions. So remember, follow the hydrogens for this kind of stuff. Oxidation means that there's no hydrogens, and then reduction means that there are hydrogens. And always try to keep an eye on what is being oxidized and what is being reduced. So how do standard reduction potentials determine the flow of electrons in a redox reaction? So you're going to have a group that is more positive and you're going to have a group that is more negative. And it would make sense that electrons want to travel. So a little electron is more negative. So it would make sense that it would want to travel towards the more positive or the larger value. But we'll cover that in detail later. What are NAD, NADP, and flavononucleotides? What do? NAD is especially important for glycolysis. You must understand the difference between NAD and NADH. So this is the oxidized version, NAD. NADH is the reduced version. So NAD will go around to different places and pick up H's or electrons, and then it will take them to the citric acid cycle so that the energy that is used to transfer electrons can be used to pump protons across the membrane. Phosphoryl group transfers and ATP. Yep, okay. Oxidation, reduction, reactions. Every oxidation, that's a loss of electrons, is accompanied by a reduction, a gain of electrons. Yep. So we just went over that. Yep. But here they're showing you little electrons, but in everything we're doing, they show up as hydrogens. So every hydrogen is attached to an electron. So we just call them electrons. Carbon exists in different oxidation states. So yeah, methane, this is the least oxidized, and then with a double bond, you get more oxidized, and you add oxygen, that's more oxidation. So the reason why is because oxygen steals the electrons. So carbon ends up with like a partial positive, and then oxygen will have a partial negative. So more reduced compounds are richer in hydrogens, like fats, and they have more electrons. More oxidized compounds have more oxygens and they have less hydrogens with less electrons. So there could be a question where they like show you a bunch of molecules and ask you like which one is the reduced form or which one is the oxidized form. Carbon exists in different oxidation states. Same thing. Oxidation, reduction, reaction. So in many biological oxidations, a compound loses 2 electrons and 2 hydrogen ions catalyzed by dehydrogenases. So D means removal, and hydrogenases, that's just like removal of hydrogens, which is the same thing as removal of electrons. So here on the left, we have lactate, we have our methyl group, we have a CH with an alcohol and the carboxylate. And then when you remove the 2 protons that leave with the electrons, so you're getting rid of this one and this one. So then that leaves behind this double bond with the oxygen. So you have to be able to follow the electrons. So from lactate and pyruvate, those molecules are almost identical, except that, you know, in pyruvate, we have the carbonyl, and in lactate, the 2 hydrogens were added and they broke up that double bond. Oxidation reduction reactions. Energy transducer can be a motor coupled to a mechanical device to do useful work. Flow on electrons in oxidation reduction reactions is responsible directly or indirectly for all the work done by living organisms. So the flow of electrons is what's super important. Source of electrons, reduced compounds. Yeah, so like the NAD that went around to pick up some hydrogens, FAD that went around to pick up some hydrogens. So those guys are the source of where we get our electrons. Food, chemical species excited by the absorption of light, photosynthetic organisms. Okay, so then right here on the left, we have anabolism. That just means making new molecules. So we use these amino acids, sugars, fatty acids, and nitrogenous bases in order to make these macromolecules like proteins, polysaccharides, lipids, and nucleic acids, and that requires energy. So making something requires energy. So you have to use ATP, NADH, NADPH, and FADH2. Now, for the opposite, after like we ate, we have to go ahead and break down. So we can break down carbohydrates, fats, proteins, and then the energy depleted end products are carbon dioxide, water, and NH3. And so when we're breaking stuff down, we actually are releasing energy from those bonds. So instead of having just ADP or NAD, the oxidized versions of these things, that's when we give them energy. So we're making ATP, NADH, NADPH, FADH. Reduction potential determines the flow of electrons. Yep. So if you guys remember the reduction potential values, whatever your value is determines where the electron is going. So always make sure that you kind of figure out who is more positive and who is more negative, and always make sure that the electron goes towards the positive. Reduction potential is a measures the affinity for electrons. So that's just like the love of electrons. So the more positive you are, the more you love those electrons. Higher reduction potential is higher affinity. Yep. So if you look at the negative sign, this is a lower number. And if something is more positive, then it's a higher number. So the higher the reduction potential, my pen is being weird. Okay, so the higher the reduction potential, the higher the affinity for the electrons. And then we have the Nernst equation. N is the number of electrons. So you're going to be given a table, and then the table is going to have the half reactions. And then in the reactions, sometimes there will be one electron, or sometimes there will be two electrons, and that's the number that you're going to use for the N. Electrons are transferred from lower to higher reduction potential. Yeah. So from lower, from the more negative to the more positive. And yeah, so the acceptor would be the more positive number. Okay, what is happening with, really? Okay. And then the donor would be the more negative. Okay. So remember, whoever has the highest reduction potential will have the more positive number, and that's going to be who's accepting that electron. And the donor is whoever has a lower number, the more negative. That's who's going to be giving away the electron. So standard reduction potential can be used to calculate free energy. So this is the equation that we learned how to use in discussion. So the standard free energy potential is equal to negative, and then the number of electrons. This is the number of electrons, and that will depend on your on your half reaction, it'll either say 2 electrons or it'll say 1 electron. The F is Faraday's constant, which will be given to you. And then the delta E is the standard reduction potential value, which is also given to you. And then the delta G, the standard delta G, what you're calculating is the energy that is made available by the spontaneous flow of electrons. So like I said, the electrons are just flowing around and they're giving you energy. So you have to calculate how much energy was given in whichever reaction. For negative delta G needs positive standard reduction potential. The acceptor, the standard reduction potential of the acceptor must be greater than the standard reduction potential of the donor. So if you have that happening, then you're going to get a negative delta G and the reaction will be favorable. So it'll move in a positive direction to the right. So this is something that I feel like this is something that might be in a multiple choice question, because Maria likes to add little things like this. I don't know if you guys have noticed. So the standard reduction potential has to be greater of the acceptor than the standard reduction potential of the donor in order for the delta G to be negative. And then here's the standard reduction potentials of some biologically important half reactions. So on the exam, if they give you a question like this, they'll probably give you like a smaller table. And then remember, so up here at the top, this is like the more positive stuff and the higher values. And then down at the bottom, there's going to be the more negative numbers and the lower values. So electrons will travel from the lower to the higher reduction potential value. Okay, and that makes more sense because electrons are negative and they want to travel to the region that is more positive. And then on the left, we just have the different types of reactions. So the one we used in lab, well, we have oxaloacetate here, fumarate, ubiquinone, iron, oxygen. Oh, okay, so if we get anything over here, these guys will be the donors, the electron donors, whoever's like more negative and then more positive, these will be the acceptors. And here's some more half reactions. So like we learned in discussion, sometimes they'll give you all negative numbers, like you'll have to choose between this and this. And then you just kind of figure out who is more positive out of the two negative numbers. So if you look up here, this is negative 0.185. That is more positive than negative 0.434. So then the electrons will move that way. Worked example, calculation of standard free energy change and free energy change of a redox reaction. Calculate the standard free energy change for the reaction in which acetaldehyde is reduced by the biological electron carrier, NADH. So we did something similar in discussion, so I'm not going to go over this. The video's already up for this. But basically, as long as you know your terminology, this will be easy for you. So I'll just go to the bottom. So standard reduction is just minus N. So again, the number of electrons. So they used 2 because in the reaction that they're using, you can see that this is 2 electrons. And then F was Faraday's constant. The reduction potential of the acceptor, which is the more positive number. So that guy right there. Minus the standard reduction potential of the donor, which we know that the donor is the more negative number, and we know that the lower, this is the more negative number. And then you just do your calculations and that's it. It's not too bad. So then you calculate the actual free energy change when acetaldehyde and NADH are 1 molar and ethanol and NAD plus are 0.1 molar. So remember, anything standard means that all of the reagents are at concentrations of 1 molar, but in reality, you can have other concentrations because we do have other concentrations of the body. That's why they have NAD plus concentration is 0.1 molar. So then this is the equation that you would use, and it's always products over reactants. So they gave you the equation up above right here. So you find, the first thing I do is find my arrow, and then I label. So on the right are my products, on the left are the reactants, and that way you know how to organize your equation. So it would be the products, ethanol and NAD plus over the reactants, and that's exactly what they have here. And then the next thing you do is just rearrange this formula to get that. Oh, there's no rearrangement. You just plug stuff in. So you just plug in, this is the answer that we just got. R, universal gas constant, T is just a temperature in Kelvin, and then ln of the product concentrations over the reactant concentrations. So once you plug everything in, there you go. Glucose oxidation in cells. So glucose, that's just a glucose molecule with 6 oxygens, gives you 6 carbon dioxides and 6 waters. So glucose oxidation has this much energy if you break all the bonds, but we don't want to break all the bonds. We want to break glucose down into pyruvate, and we want pyruvate to keep that energy in the bonds so that we can make ATP. So during the oxidation steps, the electrons are transferred to special electron carriers. And we've talked about those before, NAD, FAD. Okay, yeah, so glucose is oxidized. I could see that being, because it's very repetitive, like 3-4 lectures talk about glucose oxidation. What does that mean? It loses electrons and it loses hydrogens. So where do those electrons and hydrogens go during glucose oxidation? Well, they go to the NADH and FADH, and then they just keep going downstream until they get to the electron transport chain, which is where they donate these electrons to the different complexes there. ATP and electron carriers. So it's the same thing we went over earlier. Okay, yeah, so these are the electron carriers. Universal electron carriers, NADH and NADPH. So NADH is a nicotinamide ring. So this looks complicated, but it's really not that bad. If you look at this guy and then you look at this guy, all we have to do is look at, there's an extra hydrogen here. So that means that there's an extra electron. So this is NADH. So this is a reduced, but I guess it's only partially reduced because this right here is the fully reduced. Because there's 2 electrons. There's this guy and this guy. So there's some differences. These are water soluble coenzymes. So water soluble, they can dissociate from the enzyme after the reaction. In a typical biological oxidation reaction, hydride from an alcohol is transferred to NAD plus, giving NADH. So Maria emphasized in a couple of her lectures that these guys are water soluble and that they can dissociate. So these guys are like little Ubers. They're free to go and pick things up and travel throughout the whole city or maybe even the whole state. They are just free to just travel, and they're water soluble. So we call this dissociation. They can dissociate from the enzyme after the reaction. So I don't know if you guys remember one of your active binding sites for one of your enzymes. It was like you started off with an NAD plus, and then once the molecule, the substrate was oxidized and NADH was given that hydrogen, then it left, and then another NAD plus came in and took its place so that it could be reduced as well with one of those hydrogens. So NADHs are free to go. What else? Right here, NAD plus to NADH ratio. So the ratio of the oxidized to the reduced version is high. So there's a lot more of the NAD plus. NAD plus is used in oxidation reactions. And then over here, the ratio of NADP plus to NADPH. So again, this is the oxidized versus the reduced, is low. So that means that there's like more of this guy. So NADPH is used in reduction reactions. Okay, so these are like little facts. Like if you don't know those, then go ahead and make a flashcard for that, because I have seen that on exams before. Okay, universal electron carriers. Flavin nucleotides, FAD or FMN. So this is like the molecule by itself. And then here it is partially reduced with one hydrogen. And here it is fully reduced with two hydrogens. So hopefully the whole oxidation reduction is just becoming a lot easier for you by this point. So flavin cofactors are tightly bound. Oh, so did you notice that the NADH and NADPH were free to roam around, whereas the FAD and FMN, they're tightly bound to the proteins. So these guys are not free to roam around, they're attached. They don't diffuse from one protein to another. Standard free energy changes according to the protein to which it is bound. Can participate in one or two electron transfers. Fully reduced, partially reduced, and fully oxidized forms can be followed by absorbance as well. Okay, so yeah, so this is the fully oxidized, where there's nothing there, partially reduced, and then fully reduced. Okay, so NADH, NADPH, those guys can roam around, but FAD can't. So remember, Maria focused, she emphasized that in a couple of her lectures. Universal electron carriers, so we have NAD, NAD plus, FAD, they're all activated carriers of electrons. So I like to think of a little taxi or an Uber that just carries the electrons as passengers. And the whole reason is for fuel oxidation in the electron transport chain. Okay, so they participate in reactions such as the following. Yeah, so we're going to see this a lot in glycolysis in the citric acid cycle, where our molecule will have some hydrogens. NAD plus will show up like an empty taxi. It'll pick up its passengers, one or two hydrogens, and then it'll take off and head over to the next step. And then our substrate will be oxidized. And then this guy will be reduced. Okay, and then FADH2 participates in the following. Yep, exactly the same thing. It didn't have hydrogens, and now it does. And whenever you steal this, remember that these carbons still need to have 4 bonds. And because you took the hydrogens, they no longer do. And that's when they create a double bond. The energy conserved in NADH and FADH2 reduced electron carriers is used for production of ATP and cellular respiration. So, yep. That's the transport chain, the electron transport chain, cellular respiration. Oh no, that's right before. Okay, so let's talk about this. So stage 1 is acetyl coenzyme A production. So we can make acetyl coenzyme A from amino acids, fatty acids, or glucose. So far we've done the whole glucose. That's what we mainly studied in glycolysis. So we have to remember what this is. So, okay, so glycolysis is 10 reactions. Okay, 7 of them, or 3 of them, were irreversible because they have high negative delta G's. Okay, at the end of the 10 reactions, you end up with pyruvate. So glucose, we have to remember, was a 6-carbon molecule. 1, 2, 3, 4, 5, 6. So 6-carbon molecule, and pyruvate is a 3-carbon molecule. Go ahead and erase that. Okay, so pyruvate looks like this. You should be able to recognize the structures. So times 2. So glucose was a 6-carbon sugar, and after the 10 glycolysis reactions, you ended up with 2 pyruvate molecules. And then we had the pyruvate dehydrogenase complex. This one's really important to learn. Pyruvate dehydrogenase complex. This is a huge complex with 3 enzymes and 5 cofactors. And you can have multiples of the enzymes. For example, enzyme 1, enzyme 2, and enzyme 3, you can have 100 of each. So 5 cofactors. Okay, that's a pyruvate dehydrogenase complex. And the whole point of that is to decarboxylate pyruvate and attach it to coenzyme A. So you end up with acetyl coenzyme A, and that looks like this. We end up with the acetyl group from pyruvate, which is the methyl group, and the carbonyl, that's it. That is the acetyl group from pyruvate. And then they attach this to coenzyme A, and they attach it via coenzyme A's sulfur. Okay, so that's what that looks like. Okay, so the whole point here for stage 1 is to make acetyl coenzyme A, because pyruvate cannot enter the citric acid cycle by itself. It has to be in this form of acetyl coenzyme A. And then so you can get acetyl coenzyme A from fatty acids and also from amino acids. So then step 2, once we have our acetyl coenzyme A, we can go into the acetyl coenzyme A oxidation, which is also called the citric acid cycle. So on your final, the professors can use any of this terminology. So you have to know that acetyl coenzyme A oxidation is the same thing as the citric acid cycle. Okay, so here we start off with our 4-carbon molecule, which I covered in discussion yesterday. I covered the entire citric acid cycle in detail. So if you need more detail, go ahead and watch that video. It's already posted. But basically, acetyl coenzyme A is a 2-carbon molecule. You can see that carbon 1, carbon 2. Oxaloacetate is a 4-carbon molecule. They get together and you make citrate, which is a 6-carbon molecule. And then you continue throughout the cycle. So during the citric acid cycle, you're going to release 2 CO2s, and those are going to come from oxaloacetate. And then you're also going to be reducing a lot of these molecules. So this is a really important concept that students struggle with. So remember, so we're going to have 3 NADHs and 1 FADH2. So you see every time one of these electrons leaves, that's one of these electron carriers. So imagine a little taxi swinging by and picking up an electron and taking it to the next step. Okay, so we've got 1, 2, 3, 4 electrons. So we make 3 NADHs and 1 FADH2. But really it's called reduce. So our substrates get oxidized in order to reduce one of these molecules. And then the very last step is the electron transfer and oxidative phosphorylation. So now these guys can finally fulfill their purpose, and the whole purpose is to donate those electrons to the respiratory electron transfer chain. So by them donating those electrons, we're going to start the flow of electrons, which provides energy to pump those protons across the membrane. And then we will make, well, we'll talk about that later, but basically you also have your 2 hydrogens plus half of an O2, and that will create water. And you're going to be making ATP. Okay. So catabolism of proteins, fats, and carbohydrates in cellular respiration, the aerobic phase of catabolism by which cells consume oxygen. So anytime you see aerobic, know that oxygen is going to be the final electron acceptor. Aerobic means oxygen will be the final electron acceptor. If you do not have oxygen available to be the final electron acceptor, then you can't have aerobic respiration. It'll have to be something else, like fermentation. And you have to produce CO2 during the oxidation of fuels. Okay, so we're going to study many of these steps in the next chapters. Okay. ATP as energy currency. So this I'm going to go through pretty quickly. Basically, ATP, 3 phosphates. You use water to hydrolyze. That just means a water molecule will go and break the bond for that last phosphate. This inorganic phosphate now is resonance stabilized. So that's why it has like a lot of energy. Why is the free energy change of ATP hydrolysis so large? So that's a lot of energy that we can use, minus 30.5 kilojoules per mole. There's better charge separation in the products, yeah, because it's resonance stabilized. Stabilization of the products by resonance, greater degree of solvation, hydration of the products. ATP is thermodynamically unstable, negative hydrolysis, kinetically stable, large activation energy for uncatalyzed hydrolysis. Okay. Actual free energy change of ATP hydrolysis differs from the standard. So, okay, the actual free energy change in a process depends on the standard free energy, the actual concentrations of reactants and products. The free energy change is more favorable if the reactants concentration exceeds the equilibrium concentration. Okay, this is a fact that I think you should know, maybe make a flashcard. I could definitely see this being as one of your more challenging, like multiple choice questions. The free energy change is more favorable. That means more negative. If the reactants concentration, so remember we have reactants and then products. So if the reactants concentration exceeds its equilibrium concentration, so if this concentration is greater than its concentration at equilibrium. Magnesium partially shields the negative charges. Magnesium ATP is a true substrate for enzymes. So if you look at phosphate, look at all of those negative charges on the oxygens. It's a very highly negatively charged molecule. And magnesium has 2 positive charges that help to stabilize that. So I have seen on exams where they ask you specifically about magnesium and its role, okay? So it partially shields the negative charges. Free energy change depends on intracellular ATP, ADP, and phosphate concentrations. Under intracellular conditions, the standard free energy change phosphorylation potential in vivo is greater than the standard. If the concentration of ATP decreases, you have less amount of fuel and you have a lower phosphorylation potential. Cellular ATP is greater. So the concentration of cellular ATP is greater than the equilibrium concentration, making ATP a very potent source of chemical energy. So this is the definition from above that I told you to make a flashcard, this one. So this gave us kind of like a fact that we should know. And then the example would be ATP, because ATP cellular concentration is greater than the equilibrium concentration. And that makes it ATP a very potent source of chemical energy. And remember, KQ is at equilibrium, and then Q is the cellular concentration, like the actual physiological concentration. So these are all terminologies that could be used on your final. Remember, Q is cellular concentration or physiological concentration, actual concentrations. So you can see that there. Okay, so ATP is not the only one with high standard free energy change for hydrolysis. We have phosphoenolpyruvate, which we will see later, 1,3-bisphosphoglycerate, and phosphocreatine. So all these guys have really high negative delta G values. So PEP has minus 61. That's a lot. 1,3-bisphosphoglycerate has minus 49, and phosphocreatine has minus 43. And ATP only had like 35 point something. So hydrolysis of thioesters. So here's our acetyl coenzyme A. You can see that it has its S. And then here's an acetyl group, CH3, and a carbonyl. And then we have acetic acid. Oh yeah, so this is what the whole molecule looks like for coenzyme A. So we just don't draw it out. We only focus on this little group, the SH. And that's what we draw whenever. Okay, acetic acid, where we have the alcohol, and then we have acetate that is resonance stabilized. Minus 31.4. Ranking of biological phosphate compounds by standard free energy change. Okay. So I really like this graph because it just shows you like, so if you focus on this, we have the high energy compounds up on top and the low energy compounds on the bottom. And then the y-axis is the standard free energy change of hydrolysis. Hydrolysis is when we break that bond. So when we remove that phosphate, how much energy is released? So for ATP here, when we remove this phosphate, you can see that it's about what, 34, 35. And then for phosphocreatine, when we remove this phosphate, we can see that it's like 40 something. For 1,3-bisphosphoglycerate, when we remove this phosphate, you can see that we almost have 50 kilojoules per mole. And then for phosphoenolpyruvate, when we remove this phosphate, you can see that we have like more than 60 kilojoules per mole of energy that is released. But those are the three that we should really know. I have seen questions on an exam about like, they might give you like a multiple choice and give you the names like, oh, which of the following is not a high energy compound? And they can give you like glycerol phosphate, or glucose 6-phosphate. And then the rest of your answers would be phosphocreatine, phosphoenolpyruvate, and 1,3-bisphosphoglycerate. And you would have to choose which one is a low energy. Or they can do it backwards, give you a whole bunch of low energy, and you have to choose which one is the high energy. Phosphate can be transferred from compounds with higher standard free energy changes to those with lower. Okay, so that's an important fact to know. So This phosphate from phosphoenolpyruvate can be given to any of these molecules because it has the highest energy level. Bisphosphoglycerate right here, 1,3-bisphosphoglycerate though, that one cannot give its phosphate to phosphoenolpyruvate because phosphoenolpyruvate has a higher negative delta G, but it can give it its phosphate to ATP or to phosphocreatine. Any phosphorylated compound can be synthesized by coupling the synthesis to the breakdown of another phosphorylated compound with a more negative free energy. So this is just coupling reactions. We did do a few examples in discussion, and you basically, your first reaction, your second reaction, we learned that phosphoenolpyruvate, and you have to be able to identify those high energy compounds with their names, like PEP, that's phosphoenolpyruvate. So make little notes for yourself. So that one had a negative 61.9 kilojoules, and then ADP to make ATP, it costs 30.5. So we went ahead and coupled those reactions, and we still have negative 31.4 kilojoules left. So yeah, phosphate can be transferred from compounds with higher delta Gs to lower delta Gs. Each of the 3 phosphates of ATP, alpha, beta, and gamma is susceptible to nucleophilic attack. So yeah, you can actually break all of these, but this one gives you the most energy, the third one. How is the energy released by ATP hydrolysis used to perform work? Okay, so I'm going to skip this one because we're already 43 minutes in. I'm going to take a sip of coffee. Does anybody have questions in the meantime?
I have a quick question. When it says that the phosphate can be transferred from those with higher delta G to lower, does it mean it can be transferred from like more negative to more positive? Like higher negative? Because I was a little confused on that.
Oh, yeah, higher negative. Yep.
Oh, okay, thank you so much.
You're welcome. Let me show you the graph really quick. Yeah, this one. So on the y-axis, we have the delta G. And then, so yeah, from the highest to the lowest on this graph. So from more negative to more positive. Yep, that's a great question. Okay, so lecture.
Thank you.
Lecture 22 now. Regulation of metabolic pathways, introduction to metabolism. Okay, pathway series of related enzymatically catalyzed reactions. So they're just saying that, like, for metabolism, you usually have like enzyme 1 that performs its function, and then 2, and then 3. Okay, so cells and living organisms maintain a dynamic, steady state. Dynamic means constantly changing. Steady state is the concentrations are kept constant. So that's a definition you should know. Steady state, the concentrations are kept constant. Steady state and equilibrium are not the same thing. So the fact that they're saying that, that might be on like as a multiple choice. The concentrations in steady state might be different from the equilibrium concentrations. Remember that living cells are far from equilibrium. Yes, because otherwise we would be dead. Homeostasis occurs when concentrations of metabolites are kept at a steady state in the body. So steady state is not the same as equilibrium. So if the concentration of A increases, how can we keep the steady state? How to regulate the activity of enzymes. So I went ahead and went over this like in a lot of detail in one of the discussions, and it's been posted. But basically, enzyme regulation, the first 6 steps deal with the number of enzymes that you have. So at each of these steps is a regulation step where you can either make more of the enzyme or make less of it, or whatever you have, you can destroy it. And then steps 7 through 10 is just you have your existing enzymes and you need to modify their activity in different ways. So definitely check out the video if you want to see that in detail. So yeah, altering the number of molecules in the cell or the number of available molecules, and 7 through 10 is modulating the activity. Altering the number if it's molecules in the cell, or the number if it's available molecule, 1 through 6. So number of molecules, rate of synthesis versus degradation. Extracellular signals can be hormones, neurotransmitters, or growth factors. Gene transcription and messenger RNA translation are highly regulated. Once synthesized, protein molecules have a finite lifetime. So that means when you make something, it's not going to last forever. It's kind of like if I make a spaghetti today, it's not going to last for 50 years. It has a finite lifetime. Proteins can also be sequestered until needed. So yeah, you can have a little thing where you store your protein for a little bit longer until you need it. So average half-life in days, this means like, for example, in the liver, the proteins can last 0.9 days. That's the half-life. So really it's like almost 2 days before it's degraded. In the kidney, 1.7, so probably like 3.4 days. In the heart, 4.1 is the half-life. So the proteins in the heart last about 8.1 days. So you have to constantly be making new proteins. Modulating the activity of existing molecules, steps 7 through 10. Modulating the activity of existing molecules, enzyme, substrate. Enzyme binds the substrate. Okay, so here on the x-axis, we have ATP concentration, and on the y-axis, we have the initial velocity. So at low substrate concentration, so that's like over here, this is low substrate concentration, the change in velocity over concentration increases drastically. Yeah, so when we talked about Michaelis-Menten, we talked about that. The more to the left that you are, the lower your concentration. And if like you double that, then that's going to double your rate. But over here on the right hand side, if your concentration is really high, you can do like 1,000 times the amount of concentration, but your velocity is not going to increase. But because look, it's already like all the enzyme binding sites are already full. So no matter what, you can't increase your velocity. That's your max velocity. So, and that's what it says here. At high substrate concentration, the slope increases less. Many enzymes have a Km that is near or greater than the physiological concentration of their substrate. The concentration of ATP in animal tissues is about 5 millimolar. And then here the Vmax, half the Vmax. So the Km here, so remember the Km is the concentration at half the maximum velocity. So that's a definition you should know. The Km is the concentration at half the maximum velocity. So if we look here at half of the maximum velocity, the concentration was 5 millimolar. And that's what they're saying. ATP concentration in animal tissues is about 5 millimolar. And so for many enzymes, their Km is near or greater than the physiological concentration of that substrate. So yeah, that's like a fact. Make a little flashcard. Modulating the activity of existing enzymes. 8. Enzyme binds the ligand. Positive or negative modulator. Modulator is not the substrate. Heterotropic. Okay, so where are we? Vmax, not example of feedback inhibition, enzyme 1. Okay, so you go down here, and if you already have like 1 million Fs, then that is going to inhibit the activity of enzyme 1, because you're now accumulating your product, and you don't want to do that. So it's a good, this is negative inhibition. Okay, so it looks like this is like the middle line. So like, okay, so what we have to take from this is the 0.5. So that's the Km. Now, if your Km value moves to the right, then the enzyme becomes less effective. So that's a negative modulator. But if your Km moves to the left, then your enzyme becomes more effective. Because, I mean, so that's a positive modulator. So this makes sense because if you look here, like these lines are really steep, right? So the steeper the curve, the more effective the enzyme. Because right here we have a really low concentration of substrate, but look at the velocity here. Like that's huge. So the more to the left the Km is, the more effective the enzyme. And that would be a positive modulator. And the more to the right the Km is, the less effective. I mean, look at this slope. It's like nothing. So that's why it would be a negative modulator. Modulating the activity of existing molecules. Okay, this is 9. Phosphorylation results in altered enzymes activity. Phosphorylation is reversible. Phosphatases. So phosphatases remove phosphates and kinases add a phosphate. And where are the phosphates added? It's important. Serine, threonine, and tyrosine. Those are the amino acids that have the hydroxyl groups, and that is where the phosphate can be added. Modulating the activity of existing molecules. 10 enzymes combined with regulatory protein. Protein kinase A has 2 regulatory R and 2 catalytic C subunits. C subunit is inhibited by R subunit. Cyclic AMP concentration rises in response to epinephrine. This one's like a fact that you should probably know. I'm gonna make a flashcard. Why? Okay. Binding of cyclic AMP to R subunit activates pKa. Okay. Reactions far from equilibrium. To maintain steady state, all enzymes operate at the same rate. The net rate is equal to 10 in this example. Within a metabolic pathway, most reactions operate near to, but not at equilibrium. So this is talking about steady state, and this is in a metabolic pathway. So make flashcards of that. Key enzymes operate far from equilibrium. These are the sites of regulation. They control flow through the pathway. Okay, so then here we have, we have glycolysis and gluconeogenesis. So glycolysis moves downwards. This is breaking down glucose into 2 pyruvate molecules, and gluconeogenesis is making glucose. So they're talking about equilibrium constants, max action ratios. So remember for glycolysis, there was 3 reactions that were irreversible. That's reaction 1, and the enzyme associated with reaction 1 is hexokinase. Reaction 3, and the enzyme associated with this reaction is phosphofructokinase 1. Phosphofructokinase 1 is a highly regulated enzyme. And then here on the right, I just want to point out hexokinase, and then reaction 3 was phosphofructokinase 1. And then if we go here, this is reaction 10, and this is also irreversible. The enzyme associated with this is pyruvate kinase. Okay, so let's take a look at these 3. So this is cool. Hexokinase, it kind of tells you. There we are. Okay. And then reaction near equilibrium. So not near equilibrium, not near equilibrium, not near equilibrium. And so the delta G, if we look over here and here, so it's a highly negative delta G. So that's why those three reactions are irreversible. And in order for gluconeogenesis to happen, those reactions, you need different enzymes to bypass those reactions. So if you don't already know this stuff, you need to make flashcards. So for reaction 1, in order to bypass the activity of hexokinase, you need glucose 6-phosphatase. For reaction 3, in order to bypass the activity of phosphofructokinase 1, the super highly regulated enzyme, you need fructose 1,6-bisphosphatase 1. Now, the reason phosphofructokinase 1, one of the reasons that it's so highly regulated is because this is the first step in glycolysis where you're just like, oh, we're definitely going to do glycolysis. You can't, there's no turning back after this point. And then to overcome reaction number 10, the activity of pyruvate kinase. Okay, my pen is just not working anymore. I think, I don't know, with the Zoom, it just slows everything down and it's not, okay, it's fine. So to overcome pyruvate kinase, you need 2 enzymes. You need pyruvate carboxylase and you need PEP carboxykinase, okay? Why do reactions that are far from equilibrium need to be regulated? If these reactions are allowed to go to equilibrium, there would be chaos in the cell. Some intermediate concentrations would become as high as molar and fructose 1,6-bisphosphate, while others would drop too low to be useful, like ATP. So yeah, we can't have that. But yeah, I would make a flashcard for this. Isozymes, enzymes that differ in amino acid sequence yet catalyze the same reaction. Isozymes allow fine-tuning of metabolism. I'd make a flashcard for that. Different Km and Vmax. So isozymes are like super similar, but they have different functions. They catalyze the same reaction, but like they have different Kms and different Vmaxes. Or they respond differently to regulatory molecules. And remember, this is all for fine tuning of metabolism, isozymes. I'd make a flashcard here. They're encoded by different genes and may be expressed in a tissue specific or developmentally specific pattern. Okay. ATP and AMP are key cellular regulators. A 10% decrease in ATP Oh, it's my software that's not working. Okay. I think I just have too many documents open. Okay. So a 10% decrease in ATP leads to a dramatic increase in adenosine monophosphate, AMP. AMP can be a more potent allosteric regulator. So ADP plus phosphate is ATP, but when you work out, you use up all of your ATP and you turn it into ADP plus phosphate. Reduced nutrients supply or increased exercise, increase AMP. AMP activates AMPK, AMP activated protein kinase. Hormones help to maintain homeostasis. Homeostasis of blood glucose. You have low blood glucose, so the alpha cells release glucagon. So glucagon, you have gluconeogenesis in the liver that's making new glucose. Glucose is released into the blood and then you have normal blood glucose. So yeah, your body is always working, whether you have been starving for 20 hours or whether you are constantly eating, your body is always fixing it to where the amount of glucose in your blood remains the same. So over here, this is low blood glucose is like when you haven't eaten all day. High blood glucose is when you just ate. So then the beta cells release insulin, and then there will be peripheral tissue cells take glucose from the blood. So I would make a flashcard for both of these, glucagon and insulin. I'm pretty sure they'll probably be on the final. They're important regulators of glycolysis, gluconeogenesis, and glycogen synthesis and breakdown. So the reason for that is if you don't have enough glucose in your blood, remember that you have glycogen stores. And the reason that you have glycogen stores is for those moments where you need to break down glucose and then send it to the blood. And then if you just ate, you have tons of glucose now, and you don't want that all in your blood. So you want to make glycogen, and then you want to store it for later on whenever you're starving. Hormones help to maintain homeostasis. Glucagon and insulin bind to receptors in the membrane, leading to an intracellular cascade of events that affect protein activity. Glucose regulation, hexokinase mediated conversion of glucose to glucose 6-phosphate keeps intracellular glucose low. I would make a flashcard for this. Remember, for the process of glycolysis, there's 10 reactions. The first reaction is carried out by hexokinase. That's the enzyme, hexokinase. And what does it do? Well, you have your 6-carbon glucose. The first thing that hexokinase does is adds a phosphate to carbon number 6. And that is what keeps intracellular glucose concentrations low. And it traps it in the cell. Glucose transporters in humans. GLUT2 is the main one that we focus on in this class. Liver, you find it in the liver, pancreatic islets, the intestine, and the kidney. The KT equivalent of Km for the glucose transporter in the liver, GLUT2, is 20 millimolars. Oh, okay, this is an example, so you can go over that. GLUT4 is found in the muscle, the fat, and the heart. And the activity is increased by insulin, but GLUT2 is like the most popular one. Uptake of glucose and number of transporters. After a meal, so this is after we eat, glucose concentration in the blood increases, but the uptake of glucose from the blood to the tissues is also increased. So 1, glucose transporters are stored within the cell and membrane vesicles. So this right here is your cell. And these are just little tiny vesicles that are waiting. And these little blue things are glucose transporters. So if we don't want to import a lot of glucose into the cell, then these vesicles hide on the inner side of the cell. But number 2, when insulin interacts with its receptor, vesicles move to the surface and they fuse with the plasma membrane, and the number of glucose transporters in the membrane increases. So you see how each one of these is holding 3 glucose transporters? They were all hiding, but now we have like 15 glucose transporters whenever these guys go to the plasma membrane. That's how we rapidly take in all of the glucose away from the blood. We import it into the cells by doing this. When insulin levels drop, glucose transporters are removed from the plasma membrane by endocytosis, forming small vesicles. And number 4, the smaller vesicles fuse with a larger endosome. And 5, patches of the endosome enriched with glucose transporters bud off to become small vesicles ready to return to the surface when insulin levels rise again. So this is GLUT4 in muscle. Okay, so that was lecture 22, and we're going to quickly move on to 23. It's already 10. Okay, so glycolysis and gluconeogenesis. So glycolysis is just breaking down a 6-carbon glucose molecule into 2 pyruvates. Glycolysis, oxidation of glucose to pyruvate. So yeah, here is glucose, our 6-carbon molecule, and we break that. After 10 steps, we're left with 2 pyruvates with our methyl group, our carbonyl, and our carboxylate. So over here we have glucose, and it turns into 2 pyruvates through the process of glycolysis. And during this process, we make 1 NADH. So from pyruvate, this pyruvate can be used for different things. So if we have aerobic conditions, we go straight down and we turn those 2 pyruvates into 2 acetyl-CoAs. So this is the PDH complex with the 5 cofactors and the 3 enzymes. So here we would make 1 NADH and we would release 2 CO2 molecules. Now, if we have hypoxic, which means low oxygen, or anaerobic, no oxygen conditions, then pyruvate would take this route. So lactate fermentation, so you would make 2 lactates. So this is what happens to us if we do a lot of exercise. For example, if we're running and we use up all of our ATP, eventually we wouldn't have any more O2 to be the final electron acceptor. So we would go on lactate fermentation. That's kind of painful. So that's why it hurts when you've been running for so long. But in yeast, they can actually do ethanol fermentation from pyruvate. So now from the 2 pyruvates, that NADH that we made can head straight to the mitochondrial electron transfer chain in order to donate those electrons that it picked up. Now, from the 2 pyruvates, when we get to the 2 acetyl coenzyme A's, those are ready to enter the citric acid cycle. And here we would be left with 4 CO2s and 4 H2Os, and we would make another NADH so that it can also go to the electron transfer chain. So under aerobic conditions, NAD plus is regenerated by a mitochondrial electron transfer. So when they say regenerated, all that means is that the NADH give away the H to the electron transfer chain. And when it gives away the H, it becomes NAD plus, and then it goes back to the beginning. So that's what they mean by regenerated. So glycolysis is a sequence of enzyme catalyzed reactions by which glucose is converted into pyruvate. Yep. Its delta G is minus 85 kilojoules. A lot of the energy remains in pyruvate. So pyruvate has a lot of energy. Complete oxidation of glucose. So you have C6H12O6, that's glucose plus 6 water molecules. So I would make a flashcard with this equation if you don't have this like down. So when you have 1 glucose molecule plus 6 oxygens, you get 6 carbon dioxides plus 6 waters. So glycolysis, as we talked about, there are 10 reactions. 5 are the preparatory phase and 5 are the payoff phase. So in the preparatory phase, phosphorylation of glucose using 2 ATPs and its conversion to glyceraldehyde 3-phosphate. So you're using 2 ATPs. So I could definitely see lots of questions on the exam about this, anything to do with glycolysis. So in the preparatory phase, you are using 2 ATPs. In the payoff phase, you're using 4 ATPs. So the net ATP production for these 10 reactions of glycolysis is 2 ATP and 2 NADH. Okay, and then down here is the equation. Glucose plus 2 NAD plus 2 ADP plus 2 phosphates. And you end up with 2 pyrates, 2 NADHs, plus 2 hydrogens, plus 2 ATPs. So I would have another flashcard for this, for glycolysis. So yeah, here's the preparatory phase, the first 5 steps. So we start off with glucose, 6 carbon molecule, and then you end up with glucose 6-phosphate. That's because hexokinase just added the phosphate to carbon number 6. Okay, and in order, like, where did hexokinase get that phosphate from? If you look on the left, you can see that it got it from ATP, because ATP has now turned into ADP. Okay, so that's reaction 1, hexokinase. Just added that phosphate group to glucose at carbon number 6. Now, reaction number 2, phosphohexose isomerase. So an isomerase reaction just rearranges the molecules, and what it did is it moved the carbonyl from carbon 1 and it put it on carbon number 2. So the whole structure changes. If you're really confused about how this happened, watch the video that I have on Fisher projections, Fisher and Hawthorne, because I explain it in a lot of detail, all the rules and everything. But basically, that's all I did. The carbonyl moved from carbon 1 to carbon 2. So this was an aldose sugar, and this is now a ketose sugar. So we went from glucose 6-phosphate to fructose 6-phosphate. And then for reaction number 3, we have phosphofructokinase 1. So a kinase adds a phosphate, and now it's adding it to carbon number 1. And so where did phosphofructokinase 1 get this phosphate? Well, it got it from ATP. Okay, so we used another ATP. And so now you can see your fructose molecule, your 6-carbon sugar, and it has a phosphate at carbon number 1 and carbon number 6. Okay, so then aldolase is the next reaction, reaction number 4, aldolase. And this is the one that breaks this 6-carbon molecule into 2. So we went from fructose 1,6 bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So these molecules are really similar to each other, but we can only use glyceraldehyde 3-phosphate moving forward. So here we have our aldehyde, we have our CH with an alcohol, CH2, the oxygen, and the phosphate. So that's glyceraldehyde 3-phosphate. And for step number 5, we use triose phosphate isomerase. So we know that we're having an isomerization reaction in order to convert this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And all it does is it moves this carbonyl to this carbon over here. And that was the end of the preparatory phase reactions, the first 5. So now we're going to go ahead and start off the payoff phase reactions, the 5 through 10. So for number 6, or 6 through 10, for number 6, you'll have glyceraldehyde 3-phosphate dehydrogenase. And it's going to turn the 2 glyceraldehyde 3-phosphates into 1,3-bisphosphoglycerate. So all it does is it adds another phosphate group and it removes some hydrogens, because here we're making NADHs. We're making 2 of them because we had 2 G3Ps, or glyceraldehyde 3-phosphates. So you see these 2 NAD pluses, now they're 2 NADHs. So, and we can see that we added the phosphate. So we have phosphate here and a phosphate over here. Now for reaction number 7, we have phosphoglycerate kinase, 2 ADP, we're making 2 ATP. So they might ask you on the exam which steps in glycolysis produced ATP. That would be step 7 and step 10. We're making ATPs from step 7 and step 10. So phosphoglycerate kinase. We're going from 1,3-bisphosphoglycerate to 3-phosphoglycerate. So we removed one of the phosphates and from our molecule, and we gave it to ADP, and that's how we made ATP. So for reaction number 8, 3-phosphoglycerate, phosphoglycerate mutase. So we end up with 2-phosphoglycerate. So all that it did was it moved the phosphate from carbon number 3 to carbon number 2. That's it. And then for reaction number 9, we have enolase. And we just went from 2-phosphoglycerate to phosphoenol pyruvate. So we removed a water molecule here. And we were left behind with this double bond. So that's the enol version. And then for reaction number 10, all we did was remove that phosphate. So pyruvate kinase removed that last phosphate, and that's why we made ATP. We gave it to ADP to make ATP. And then our end product is pyruvate. So that's how the 10 reactions of glycolysis work. The steps that require ATP are steps 1 and 3, and the steps that produce ATP are 7 and 10. Okay, looks like we have some questions. So okay, if you I don't know that I'm gonna go into that much detail, but let me point you in the right direction. Let's see here. Here we go. Okay, if you look at discussion, I forgot what discussion this is. Okay, just look at this discussion, whichever one it is. So this is the one I covered a couple of weeks ago, and I annotated it like super good for you. So I'm not going to go over any of this stuff today, but basically you have glucose, glucose 6-phosphate, and then right here it tells you the type of reaction. So because a phosphate was transferred, this is a phosphorylation reaction, and you should know it. Reaction number 2, isomerization reaction. Reaction number 3, phosphorylation reaction. So yeah, you have that information already. Okay, where was I? Was I at 22 or 23? Okay. You are welcome. Let's see. So lecture 20, I don't think it was on the final. And if it is, I don't think we'll have time to cover it today. I started with 21. So net gain of ATP is 2 or 4 because there's 2 pyruvate. Net gain would be 2, I believe. Yeah, so follow the formula down here. So 2 ATP, that would be the net gain. All right, what else could they possibly ask you? Let me see, I have a set of notes. Let me turn on the light. Okay, so you should be making notes of which ones of these are your high energy molecules. So phosphoenol pyruvate, if you remember your little table that I showed you with the high energy molecules on top, phosphoenol pyruvate is one of those high energy molecules. Okay, so I'm going to keep going. Chemical logic of glycolysis. So I have a video where I cover this in detail, but it's basically what I just went over. They just have it in the linear format. Free energy changes during glycolysis. Yeah, so here's reaction 1, 3, and 10 with negative delta G values. So those are the irreversible reactions. And then this equation, you should be familiar with it, just like how to work with it. The RT ln of Q. Remember, Q is just the concentration of the products over reactants under physiological conditions. So glycolysis, the preparatory phase. So step 1, phosphorylation of glucose. Yep, we learned that this is hexokinase and it just adds a phosphate to glucose number to carbon number 6. So this is a phosphorylation reaction. Now they tell you like the reasons for this. The first thing that hexokinase does is phosphorylate in order to trap glucose inside the cell. And it also lowers the intracellular glucose concentration to allow further uptake. Yeah, if we didn't phosphorylate, then the cell would be like, oh, you have too much glucose in there. We don't need to uptake anymore. And we don't want that to happen, because if that happens, then it'll just end up in your blood. And we don't want that. That's the whole point. We're trying to regulate blood glucose levels. So that's why it gets phosphorylated, trapped, and the cell continues to take in more and more glucose. So phosphohexose isomerase, this is step 2. It makes the next steps easier. Carbon 1 of fructose is easier to phosphorylate. Oh yeah, so this is just where they changed the carbonyl. So if you look here at glucose 6-phosphate, the carbonyl was at carbon 1, so this was an aldehyde sugar, and then we moved it to carbon number 2. Wait, how come they didn't do that here? Ugh. Okay, just look at this guy right here. So you can tell that this is a fructose sugar because now the carbonyl group is on carbon number 2. So you see here, this was the anomeric carbon 1 on glucose 6-phosphate, and you can see how the O is right there. That's the carbonyl on carbon number 1. But if you look at the fructose 6-phosphate, carbon number 1 is up here, CH2OH. So remember to watch that video if you don't know how this happened, because it could take me like 30 minutes to explain that. But you see carbon 2 here, and now that oxygen is attached to carbon 2, so that's where the carbonyl is, and that's why this is a fructose. So this was the second enzyme. And the reason that we're moving the carbonyl group is so that later we can go ahead, there needs to be an alcohol at the end so that we can phosphorylate. Because remember, the whole point is that we need to phosphorylate both ends of the carbon, of the 6-carbon sugar, in order to move downstream for the glycolysis reactions. So mechanism of phosphohexose isomerase. Okay, so you start off with glucose that has been phosphorylated at carbon number 6. You get binding and opening of the ring. So the ring opens. You can go ahead and count your carbons, 1, 2, 3, 4, 5, 6. And you can see your aldehyde here. So what happens is that you get a proton abstraction by active site glutamic acid. So that guy right there. It leads to a cis enediol formation. So this guy basically wants to steal that proton, and it does. And then that leads to that double bond within the carbons. And then you get general acid catalysis by the same glutamic acid facilitates formation of fructose 6-phosphate. So now basically this bond will steal the hydrogen back. Then you have general, oh, okay, I already said that. Dissociation and closing of the ring. So now the carbonyl moved. You can see that it moved because these electrons that were on this proton went over here. So then this guy stole the hydrogen, and that's how that hydrogen ended there. But when these electrons jumped over here, that's how that carbonyl ended here on carbon number 2. So if you count the carbons, carbon 1, now there's an alcohol there, which is what we wanted, because now for the next steps, we can go ahead and phosphorylate that section. And then we'll have a phosphate at carbon number 6 and carbon number 1, which is what we want. And you can see that the carbonyl is at carbon number 2, so it's not in the way anymore. And then once that happens, this guy is free to go. So that's phosphohexose isomerase. Step 3, second priming phosphorylation. That's phosphofructokinase 1. Okay, so this is a really highly regulated enzyme. This is reaction number 3 of glycolysis. So you should know everything about this enzyme. So what is its function? Well, we had fructose 6-phosphate. That's the substrate. So there was a phosphate at carbon number 6, and we already went ahead and moved the carbonyl to carbon number 2. And so at carbon number 1, we have an alcohol group now. So that's this guy right here. So phosphofructokinase 1 is going to add a phosphate group to carbon number 1. So now we're going to have a phosphate at carbon 6 and a phosphate at carbon 1. So this is fructose 1,6 bisphosphate. So you allow for 2 3-carbon sugars with 1 phosphate each after step 4. So yeah, the whole reason we wanted to add a phosphate to each of these is because we're going to cut this molecule in half, and each half has to have its own phosphate. So the reason this phosphofructokinase enzyme 1 is so highly regulated is because it is the first committed step of glycolysis. Okay, and then they tell you what bis and di is. So bisphosphate is 2 separate monophosphoryl groups are present, and di is 2 phosphoryl groups are present and connected by an anhydride bond. So this is highly thermodynamically favorable. Phosphofructokinase 1 is highly regulated. Do not burn glucose if there's plenty of ATP. Step 4, cleavage. Okay, so I went over this in the video, so I'm just going to skip. Under what conditions encountered in normal cell will the free energy change be negative, enabling this reaction to proceed to the right? Okay, so the answer for this was B, I believe. The reaction will not go to the right spontaneously under any conditions because the standard free energy change is positive. No. Okay, I'm lost. So we're just going to skip this slide because I don't remember. Mechanism of class 1 aldolase. Okay, so your professor does seem to care a lot about the mechanisms of some of your enzymes. So I'm just going to cover those mechanisms. So mechanism of class 1 aldolase. So we start off by fructose 1,6 bisphosphate. So here we have the 2 phosphates that we just added. So active site lysine. So now we have a lysine. It attacks the substrate carbonyl, leading to the formation of a tetrahedral intermediate. So you can see that lysine attacks that carbon. Those electrons move. And then step 2. Rearrangement leads to the formation of the protonated shift base on the enzyme, and you get electron delocalization, and that facilitates the subsequent steps. So you basically just follow the protons, but it's reorganization. And then for step 3, you have a carbon-carbon bond cleavage. And so that was the whole point here, is to break that 6-carbon molecule into 2 3-carbon molecules. This is what this enzyme is doing, this aldolase. It's breaking that 6-carbon molecule. Now that it has the 2 phosphates, 1 at each end, it's just rearranging itself so that it can be cleaved. And so when it is cleaved, you release the first product, which is glyceraldehyde 3-phosphate, and that's this bottom half right here. So here's carbon 1, carbon 2, carbon 3. So carbon 4, 5, and 6 are released as glyceraldehyde 3-phosphate. And then you have an isomerization reaction. So again, rearrangement. And then for step 5, we have a shift base is hydrolyzed. So that's when water comes in and to break the bond in the reverse of shift base formation. So water comes in to break that bond to release this guy, and now it's set free. Dihydroxyacetone phosphate, it's set free. So the aldolase is just how this enzyme active site breaks that 6-carbon molecule into the 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 5, triose phosphate isomerase. So that's the interconversion. So I would make a flashcard on this. I have seen many questions like on finals about this enzyme or this material. Triose phosphate isomerase. So remember, we have dihydroxyacetone phosphate and we have glyceraldehyde 3-phosphate. Those are the 3-carbon sugars that were broken from the 6-carbon molecule. So this triose phosphate isomerase, it can actually turn this G3P, this glyceraldehyde 3-phosphate, into dihydroxyacetone phosphate, or it can turn dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. It's important to remember that it can go in either direction. It can go backwards. We're so used to just thinking about the fact that it turns dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, because these guys can move to step 6 of glycolysis, but dihydroxyacetone phosphate cannot move to step 6 of glycolysis. But sometimes if we already have a lot of ATP, then this enzyme could be like, oh, we already have a lot of ATP. Never mind, let me go back. So instead of turning dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, it can go backwards and go, okay, we're going to turn all the glyceraldehyde 3-phosphates into dihydroxyacetone phosphates. So allow glycolysis to proceed to payoff phase by a single chemical pathway. Only glyceraldehyde 3-phosphate, only GAP is the substrate for the next enzyme, dihydroxyacetone phosphate. So know these acronyms for those molecules and enzymes. They must be converted to GAP by triose phosphate isomerase for reaction number 6. So yeah, this is just the 6-carbon molecule with the 2 phosphates, and how when they're broken down by aldolase, you have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and you can see that's like the carbons that they come from. So I have seen questions like in multiple choice, like, oh, which 3 carbons from the original glucose molecule make up dihydroxyacetone phosphate, and it's carbons 1, 2, and 3. Which 3 carbons make up, from the original glucose molecule make up G3P? Oh, it's 4, 5, and 6. Now, pyruvate, or what is here, derived from glucose carbons. Okay, yeah, so this, we'll talk about it later. Glycolysis, the payoff phase. So now it's steps 5 through 10. So oxidation and phosphorylation, first ATP forming reaction, substrate level phosphorylation. Oh, okay, so every time you make ATP, you actually have to know how it was made. So for step 7 and 10, we made ATP, and both times it was made by substrate level phosphorylation. Okay. So if you take a look here, you can see that the substrate had 2 phosphates. We took one and gave it to ADP, and that's how we made ATP. So that's substrate level phosphorylation. Okay, and I already covered these. Okay. Okay, so step number 6, oxidation of GAP. So GAP, glyceraldehyde 3-phosphate plus inorganic phosphate. And you have the enzyme glyceraldehyde 3-phosphate dehydrogenase. And you end up with 1,3-bisphosphoglycerate. So you added a phosphate to carbon number 3. Oh no, we already had a phosphate on carbon number 3. We are adding a phosphate to carbon number 1. So you see that's carbon number 1 here. And you end up with 1,3-BPG. So remember, we already talked about phosphoenol pyruvate and how that's a high energy molecule. But here in step 6, this is also a high energy molecule. We have 1,3-bisphosphoglycerate. So definitely know that. So this allows for net production of ATP via glycolysis. So remember, this is a lot higher on the scale of energy, because it's like a -65 delta G, it looks like. Oh no, for this one it's like, I don't know, like almost 50. So it can definitely be used to make ATP. Okay, and then oxidation of aldehyde with NAD plus gives NADH. So yeah, in addition to this glyceraldehyde 3-phosphate dehydrogenase from step 6, this enzyme, in addition to making this high energy molecule 1,3-BPG, it also reduces NAD plus into NADH. And we already know that that's going to head over to the citric acid cycle to create ATP. So here's another mechanism. This one is GAPDH, glyceraldehyde 3-phosphate dehydrogenase. So we start off with NAD plus. And like we learned earlier, NAD plus and NADPH, those guys are free to come and go. So this one is sitting here now, but once it picks up its hydrogens, it can take off and be replaced with a different one. Whereas if you had FAD, then that one would be stuck to the enzyme. It would not be able to just travel and take off. Okay, so, glyceraldehyde 3-phosphate, formation of enzyme substrate complex. The active site cysteine has a reduced pKa of 5.5 instead of 8. Okay, so pKa, let's refresh our minds. The pKa is the pH at which that group will donate its proton. So normally cysteine does not donate its proton until the pH is 8. But here, because of its local environment, it has been affected in a way that the pKa has been dropped to 5.5. So at physiological pH, which is like 7, it definitely has donated its proton, and that's why it has an S with a negative. It does not have a hydrogen. Okay, that's why. So now when G3P, glyceraldehyde 3-phosphate, comes in, the sulfur attacks a covalent thiohemiacetal linkage forms between the substrate and the sulfur group of the cysteine residue. So they form a bond. And then step number 3, the enzyme substrate intermediate is oxidized by the NAD plus bound to the active site. So you can see that this NAD plus, right, it wants to steal that hydrogen. So the electrons move from here and then they go there, and then so NADH. So it did steal that hydrogen. So now it can take off. And we already know that whenever a hydrogen is stolen from a carbon, you normally end up with a double bond there in that location. And then the NADH product leaves the active site and is replaced by another molecule of NAD plus. And then the covalent thioester linkage between, so whenever they say thioester linkage, they're just talking about this carbonyl that is attached to this sulfur on the cysteine. That's a thioester linkage. So we need to detach it. So it undergoes phosphorylysis. So it's attacked by the phosphate. And it releases the second product as 1,3-bisphosphoglycerate. So notice that oxygen attached this carbon. And so that's how we got that second phosphate at that location. And then it was able to detach from the enzyme, because remember this blue thing right here is the enzyme. And so now we have 1,3-bisphosphoglycerate with this phosphate attached to carbon number 3 and this phosphate attached to carbon number 1. Okay, so that was the mechanism for GAPDH. Step 7, the first production of ATP. Okay, so again, we have our high energy molecules, phosphoenolpyruvate and 1,3-bisphosphoglycerate. Both of these can be used to make ATP because it's down here. And here we're talking about 1,3-bisphosphoglycerate. This is substrate level phosphorylation because our substrate is 1,3-bisphosphoglycerate. We're stealing the phosphate from it and we're giving it to ADP in order to make A3P. So 1,3-bisphosphoglycerate is a high energy compound and can donate the phosphate group to ADP to make it ATP. Kinases are enzymes that transfer phosphate groups between ATP and substrates. Reversible because it is coupled to GAPDH. So phosphoglycerate kinase is reversible. Step 8, migration of the phosphate. Okay, so this is a mutase. Okay, so basically 3-PG, that's 3-phosphoglycerate. This is carbon 1, 2, and 3. And we have the phosphate group at that location because the name tells you where the phosphate is. So this phosphoglycerate mutase, so all that it does is it changes the phosphate from carbon number 3 to carbon number 2. And this helps to form a high energy phosphate compound in the next step. Mutases catalyze the apparent migration of the functional group. Step 9, dehydration of 2-phosphoglycerate to phosphoenolpyruvate. So here in step 9, we use enolase and we're removing a water molecule. So if you look at this 2-phosphoglycerate, that's what we had earlier. We had our CH2 with an alcohol. We had our carbon where we just added this phosphate group to, we just moved it from 3 to 2. There's a hydrogen there, and then this. So we're removing a water molecule. So that's 2 hydrogens and the oxygen. So this is where the water molecule came from. And so then we end up with phosphoenolpyruvate. So because this is in the enol form, and we have the phosphate in the middle, this is a phosphoenolpyruvate is one of our high energy molecules. Generates a high energy phosphate compound. PEP is used to generate ATP in the next step. And then here they show you the enol form and then the keto form. And then step 10, this is the last step in glycolysis. So we end up with pyruvate. So here is PEP, phosphoenolpyruvate, plus ADP. So that tells you we're going to make ATP via substrate level phosphorylation. So we're taking this phosphate from our substrate and we're adding it to ADP to make ATP. And you can see that there's magnesium and potassium to help stabilize the negative charges. So substrate level phosphorylation, net production of 2 ATP per glucose. So yeah, this is happening twice. So that's why there's 2 ATPs. This is highly thermodynamically favorable and irreversible. This is regulated by ATP, divalent metals, and other metabolites. So this is step 10. So remember, in glycolysis, steps 1, 3, and 10 have really high negative delta G values, and they are irreversible. Chemical logic of glycolysis. I went over that in discussion in detail. What is the net gain of glycolysis? 2 ATPs. Because it costs 2 in the preparatory phase, and then you made 4 in the payoff. So 4 minus 2 is 2. What would happen to glycolysis if there is no more NAD+? So this is an important question. I think she might talk about it in the final. Under anaerobic conditions, glycolysis stops until NAD+ can be replenished by fermentation. So if you don't have NAD+, you can't continue with glycolysis. So fermentation is a reduction of pyruvate to another product. So we talked about that earlier from pyruvate, how you can go to 2-lactate or to ethanol, depending on what you are. So lactate fermentation in humans and ethanol fermentation in yeast. Regenerates NAD+ for further glycolysis under anaerobic conditions. So yeah, if you undergo fermentation, lactate fermentation, you can see that an NADH is turned into NAD+. Generation of energy, ATP, without consuming oxygen or NAD+, no net change in oxidation state of the sugars. The process is used in the production of food from beer to yogurt to soy sauce. Pyruvate decarboxylase and alcohol dehydrogenase reaction. Oh, I covered pyruvate decarboxylase in the most recent video for discussion. Animals undergo lactic acid fermentation. Yeah, so this is what I was talking about when you're running and how like it hurts. You start off with pyruvate. Here's your methyl, your carbonyl, and then you have your NADH that turns into NAD plus. The enzyme is lactate dehydrogenase. And remember, this is when there's no oxygen. So this is anaerobic respiration. And here's L-lactate. So the hydrogen was given to us by NADH. And that's why the double bond from the carbonyl was broken. And now you have an alcohol here and a hydrogen here. So normally we're used to our substrate giving the hydrogens to NAD plus to make NADH. But in this case, it's backwards. Yeast undergo ethanol fermentation. So in discussion, there's a question about this, and it says like, oh, let me just read this. Two-step reduction of pyruvate to ethanol. So 2 steps, there's 2 enzymes. Humans do not have pyruvate decarboxylase, so we don't have this enzyme. That's why we don't produce our own alcohol. We do express alcohol dehydrogenase for ethanol metabolism, but it is largely used in the reverse reaction. CO2 produced in the first step is responsible for the carbonation in beer. So I have seen that like on exams, like, oh, where does the carbonation in your beer come from? Well, it comes from this enzyme, pyruvate decarboxylase. When it removes that CO2, boom, there you go, CO2. And it's also responsible for the dough rising when we bake bread. Both steps require cofactors. There's no net change in carbon oxidation. What is thiamine pyrophosphate? TPP. So that is one of the 5 cofactors in the pyruvate dehydrogenase complex, TPP, and it helps to decarboxylate the pyruvate. So it helps to remove that CO2. Glycogen, starch, disaccharides, and hexoses enter glycolysis. Oh yeah, so it's just saying that like it's not, there's other things that can go in. Like glycogen, remember that's our glycogen stores. So we can have like thousands of glucose monomers stored as little granules inside cells, and then they come out when we need glucose. Well, starch is for plants, disaccharides, that's 2 sugars, and hexoses, that's the 6 carbon sugars, they enter glycolysis. Okay, so we're moving on to lecture 24. If anybody has questions, put them in the chat. I need a 2-minute break really quick. I don't know. She specified that you should not memorize things, but she wants you to understand. But then on the exam, like you do have to know and be able to identify, like, I don't know, like I don't know if she'll go as specific as like the amino acids that are involved. Maybe more the overall concept, like the aldolase. The whole point was to break that 6 carbon molecule into 2 3 carbon molecules. The more you know, the more prepared you will be. So it depends on each student, like how well you have studied to this point. How much time do you realistically have? So yeah, that'll just depend on each student. Third regulation point, pyruvate kinase. Oh wait. Am I on 23 now? Or am I on 24? Sorry, I don't know which one I just finished. Okay, I don't think I've started gluconeogenesis. So we'll start 23 right now. Gluconeogenesis, making new glucose. So pathway that converts pyruvate and related 3 and 4 carbon compounds to glucose. Physiologically necessary. Glucose is the major fuel source for human brain. I've seen that on exams that our brain really needs glucose. The nervous system and red blood cells. It allows generation of glucose from non-carbohydrate precursors when glycogen stores are depleted. So if we have already used all of our glycogen, then we can go ahead and start using other things that are not carbohydrates in order to make glucose. And this is like when you're starving, when there's vigorous exercise. You can generate glucose from amino acids, but not fatty acids. So make a flashcard. Gluconeogenesis, making new glucose, is expensive, but it's necessary. So these are the things that you'll need. You'll need 2 pyruvates, 4 ATPs, 2 GTPs, and 2 NADHs. Those 2 hydrogens and 4 water molecules. So I make a flashcard, and then what do you get in return? You get glucose, you get 4 ADP, you get 2 GDP, 6 phosphates, and 2 NAD plus. In mammals, gluconeogenesis occurs mainly in the liver. Animals lack the enzymatic machinery to convert acetyl-CoA from fatty acids to pyruvate. Plants and microorganisms have the glycosylate pathway, which allows them to make glucose from fatty acids. Okay, so plants can, but we cannot. From fatty acids. Okay. Glycolysis versus gluconeogenesis. Okay, so reversible reactions are used by both pathways. So out of the 10 reactions from glycolysis, 7 of those are used in both pathways, glycolysis and gluconeogenesis. But 3 of them are irreversible in glycolysis. So those have to be bypassed using different enzymes in gluconeogenesis. There's no ATP generated during gluconeogenesis, no, because this gluconeogenesis process costs money, costs energy. Different enzymes in the different pathways differentially regulated to prevent a futile cycle. Okay, so the most important thing to learn here for glycolysis versus gluconeogenesis is everything we just covered. So you should definitely know, like, from glucose to glucose 6-phosphate. In glycolysis, that enzyme was hexokinase, but in gluconeogenesis, it's glucose 6-phosphatase. So for the reactions 1, 3, and 10, it's really important. Steps 1 and 2, pyruvate and phosphoenol pyruvate. So pyruvate to PEP. Glycolysis happens in the cytosol. Pyruvate to oxaloacetate happens in the mitochondria. You do have to know where things happen. Carbon is added and immediately removed, activation by CO2. Okay, so you have bicarbonate plus pyruvate, and then you have pyruvate carboxylase. It uses ATP, and biotin is a carbon dioxide carrier. They might have something about that. So then you end up with oxaloacetate, which is that 4-carbon molecule. And then you have GTP. So you steal the last phosphate. So instead of using ATP, you're using GTP to steal the last phosphate. And a PEP carboxykinase will go ahead and make phosphoenolpyruvate, that really high energy molecule. So pyruvate is converted to oxaloacetate in mitochondria. Okay, so I hope you see what's happening here. So for glycolysis, right here, glycolysis, there was 10 reactions and we ended up with 2 pyruvates. So from these 2 pyruvates, they're working their way backwards. So they're showing you backwards. Okay, now that we have pyruvate and we're going to stop the process of glycolysis, we actually need to start the process of gluconeogenesis. So we're going to start making glucose from pyruvate. So you can see that they're using these enzymes. So the first enzyme is pyruvate carboxylase, and the second one is PEP. That's phosphoenolpyruvate carboxykinase. So that's what's happening. And there it is, phosphoenolpyruvate. So pyruvate is converted to oxaloacetate in the mitochondria. Oh, I'm on 24? Yeah, I'm on 24. Thank you. I was super lost. Okay. So NADH, NAD plus, 8 times 10 to the negative 4. NADH is consumed during gluconeogenesis. Okay, so this is important. Remember during glycolysis how we made NADHs, but here in gluconeogenesis, we're using NADHs. Transport of malate to the cytosol and later oxidation help move the NADH to the cytosol. Okay, so there is no oxaloacetate transporter, but there is a malate transporter. So that's kind of an important fact to know. Here in the mitochondria, we don't have a transporter for oxaloacetate, but we need to get oxaloacetate out of the mitochondria. So in order to do that, we have to first convert it to malate. And once malate is outside, we reconvert it to oxaloacetate. Okay, so we start with pyruvate that goes into the mitochondria from the cytosol, and then we use pyruvate carboxylase. Okay, so CO2 comes in and ATP comes in, and we make oxaloacetate, and then mitochondrial malate dehydrogenase turns oxaloacetate into malate using an NADH. So you can see that during gluconeogenesis, we're just using a bunch of stuff. And that makes sense because we're making something. We're making glucose. So pyruvate in the cytosol to pyruvate in the mitochondria turns into oxaloacetate. That turns into malate. And then the malate, we do have a transporter protein. So malate can be transported into the cytosol. Here in the cytosol, we use NAD plus and we turn it into NADH. So finally in the cytosol, we're making an NADH. We're using cytosolic malate dehydrogenase. So we have 2 types of malate dehydrogenase, one in the cytosol and one in the mitochondria. And this one turns this into oxaloacetate. And then we have cytosolic PEP carboxykinase. So this releases a CO2 and we end up with phosphoenolpyruvate in the cytosol. But yeah, it's important to pay attention to that because the professor might ask you, like, how do you get oxaloacetate out of the mitochondria if you do not have a transporter? Okay, so you have to convert it into malate. You export malate and then you reconvert it to oxaloacetate. Lactate produced in muscle and erythrocytes. All right, so we have lactate here from muscle and erythrocytes. You have an NAD plus that turns into NADH. So we're making an NADH using lactate dehydrogenase. So we go from lactate to pyruvate, and now we know that pyruvate can enter the mitochondria. Okay, and then here in the mitochondria, we use CO2 and we use an ATP, and the enzyme pyruvate carboxylase turns pyruvate into oxaloacetate. And then from oxaloacetate, we use a GTP and a CO2, and mitochondrial phosphoenolpyruvate carboxykinase turns oxaloacetate into phosphoenolpyruvate. And now phosphoenolpyruvate can leave the mitochondria into the cytosol. So additional bypasses. So this is reaction. Let me see if I can write. So this was reaction 1. No, it doesn't let me. I may have to just restart my entire computer. Okay, I guess I'll just try to keep going without writing. Okay, this is reaction 1, and this is reaction 3, and this is reaction 10. So we just went over reaction 10 using pyruvate carboxylase and phosphoenolpyruvate carboxykinase. So we went over that, how gluconeogenesis has to use those two enzymes to bypass reaction number 10 and to go from pyruvate to oxaloacetate to phosphoenolpyruvate. Okay, so now we're talking about additional bypasses. So fructose 1,6 bisphosphate, fructose 6-phosphate, 2-fructose 6-phosphate. So here, down here, we have fructose 1,6 bisphosphate. So this was a fructose molecule that had a phosphate at carbon number 1 and a phosphate at carbon number 6. And we need to move backwards to the fructose that had only the phosphate at carbon number 6, but not at carbon 1. So in order to do that, we got to use this enzyme fructose 1,6 bisphosphatase 1. So it's removing the phosphate from carbon number 1. So, and then the other one is glucose 6-phosphate. So here we're starting with glucose that has the phosphate at carbon number 6, and glucose 6-phosphatase, that enzyme is removing that phosphate from carbon number 6, and you end up with glucose. So both phosphatases, because they're both removing a phosphate, they catalyze the hydrolysis reactions, and no ATP is generated. Nope, because we learned that in the process of gluconeogenesis, we're not making any ATP. So glucose 6-phosphatase only in the liver, kidney, and epithelial cells of the small intestine. Only tissues can export glucose. That might be important. So glucose 6-phosphatase, that's this guy right here, this first enzyme. The enzyme that bypasses reaction number 1 in glycolysis, glucose 6-phosphatase, is only found in the liver, the kidney, and the epithelial cells of the small intestine. Glycolysis and gluconeogenesis are reciprocally regulated. Under unilateral conditions, overall, the standard or the energy change in glycolysis is -63, and the energy change in gluconeogenesis is -16. So it doesn't okay. So that's kind of like really different. To form one glucose molecule, gluconeogenesis uses 6 high-energy phosphate groups, 4 ATPs and 2 GTPs, and 2 molecules of NADH. So this is, I can almost guarantee it's going to be on the final. She's definitely going to be asking you like differences between glycolysis and gluconeogenesis. So to make one glucose, you need 6 high energy phosphate groups. So ATP is a high energy phosphate group, and so is GTP. So you need 4 ATPs and 2 GTPs for this gluconeogenesis pathway. You also need 2 molecules of NADH. So in glycolysis, as you move down glycolysis, you're making ATP in steps 7 and 10, and you're making NADH. But in gluconeogenesis, you are using 4 ATPs, 2 GTPs, and 2 NADHs. So both processes are essentially irreversible in cells, and they are reciprocally regulated to prevent wasteful operation of both ways at the same time. So reciprocally regulated, that means that we keep a constant regulation of both at the same time. And there's certain molecules that will have opposite effects. Like they'll help promote one pathway while inhibiting the other pathway. You don't want them both working at the same time because that would be wasteful. So glycolysis and gluconeogenesis are reciprocally regulated. So now they're going to talk about that here. So this is reaction, where are we? Fructose 6-phosphate to fructose 1,6 bisphosphate. So this is reaction 3. So this is one of those high delta G that is negative, so it's irreversible. So phosphofructokinase is the enzyme for glycolysis, and fructose 1,6 bisphosphatase is the enzyme for gluconeogenesis. So phosphofructokinase adds a phosphate to carbon number 1, but fructose 1,6 bisphosphatase removes that phosphate from carbon number 1. So phosphofructokinase is the most important control. So I've been telling you for like 3 lectures now, phosphofructokinase 1 is a very highly regulated enzyme because it's the first committed step in glycolysis. It's step number 3 and it's irreversible. So I would say that these 2 enzymes are really important, phosphofructokinase and fructose 1,6 bisphosphatase. So another important thing to recognize and kind of like try to understand is everything in green will activate or promote that pathway. So for glycolysis, we're going downwards, and for gluconeogenesis, we're going upwards. So for example, citrate promotes the process of gluconeogenesis, but at the same time, it inhibits the process of glycolysis. So how does it do this? Citrate promotes fructose 1,6 bisphosphatase. Citrate inhibits phosphofructokinase 1. So now, adenosine monophosphate is another one. So adenosine monophosphate means that you have low ATP because this is AMP. There's only one phosphate. You like the molecule that has the 3 phosphates, so you can use those for energy. When you use up all the energy, you're left with AMPs. And so if you have low energies, that means that glycolysis is going to be promoted, and that's why it has a little P, a little plus sign, and it's green. So if you have low energy in your blood or in your body, you're going to promote the process of glycolysis and you're going to move down this way. Because glycolysis, the whole purpose is to create ATP. Gluconeogenesis, that creates new glucose, but it uses ATP. And if you don't have a lot of ATP to start with, you can't be wasteful and do the process of gluconeogenesis. You have to first make ATP. So I hope you, and then so AMP, like if you have a lot of it, it inhibits this pathway. So you can see that AMP promotes glycolysis, but it inhibits gluconeogenesis. And that's how things are highly regulated. Does it use GTP if you make it into malate instead of directly into PEP? I don't know. I don't know that I want to go back because we're already short on time. Yeah, anything I can't answer, I would say just go to the professor's office hours. Okay, where are we? Regulation of glycolysis in muscle. Okay, so at rest and then during exercise. So let's look at the at rest. At rest glycolysis is not very active. Then arrows. Yeah, so you're not breaking down glucose at rest. The high concentration of ATP inhibits phosphofructokinase, pyruvate kinase, and hexokinase. Glucose 6 phosphate is converted into glycogen. So here they're just using the names of things. So you should have the process of glycolysis like down, like you should know all the enzymes and everything, so that when you see stuff like this on the exam, you're not stressed out. You already know. So basically a lot of ATP will inhibit through these three enzymes: hexokinase, phosphofructokinase, and pyruvate kinase. And then during exercise, glycolysis is stimulated. So we have to think about this logically, and this is what Maria is really reinforcing. She's saying they have to be able to connect the dots. So during exercise, you have worked out and you have used up all of your ATP. So you need to make more ATP. So which of the two processes is used to make ATP? Glycolysis, right? So then glycolysis is stimulated during exercise. So you start off with glucose. Okay, low energy AMP will stimulate phosphofructokinase and feed forward stimulation of pyruvate kinase. So during exercise, the decrease in the ATP to AMP ratio resulting from muscle contraction activates phosphofructokinase and glycolysis through the thick arrows. Okay, so that makes sense. That was the enzyme that we just talked about for reaction number 3. So we just said AMP right there with a positive, with a green. It stimulates glycolysis because it's low energy, but at the same time, AMP inhibits this enzyme for gluconeogenesis. First regulation point is hexokinase. So we know that hexokinase is the one that for the first step in glycolysis and where you go from glucose to glucose 6 phosphate. So when you have a lot of glucose 6 phosphate, there's a negative feedback and hexokinase is inhibited. Muscle consumes glucose for energy production. Hexokinase is inhibited by glucose 6 phosphate. In the liver, maintains blood glucose homeostasis, and it consumes or produces glucose depending on the conditions. So I really think that Maria is probably going to have like questions about what is happening in the liver versus what is happening in muscle cells. So I would have like a little chart and I would put liver and muscle, and anything that I encounter throughout my studies, I would just make little notes to just refresh your memory right before the exam, because there might be a question about stuff like that. So the first regulation point, hexokinase. So remember, the regulation points are at the irreversible reaction. So like reaction 1, reaction 3, and reaction 10. So hexokinase has 4 isozymes. And so hexokinase 1 is expressed in all tissues to different levels. Hexokinase 4, glucokinase, is only expressed in the liver. So that's an important fact. It has a higher Km. So what did we learn about Km? If the Km moves to the right, that means that you have a higher Km. That means that your enzyme efficiency, it goes down. So responsive to higher glucose concentration. So not inhibited by glucose 6 phosphate, so can function at higher glucose concentrations. Functions to clear blood glucose at higher glucose concentration for storage as glycogen. So the reason, now they're telling you the reason that it has a higher Km. So it has a higher Km and it makes it less efficient because it's able to just take up like a bunch more glucose. Because the whole purpose of it of the liver is to store your glucose, right? You want to remove it from the blood as soon as possible because you don't want to have high glucose in your blood because that can lead to disease. So hexokinase 4 in the liver is regulated. Glucose competes with fructose 6-phosphate for binding. When glucose binds, it causes dissociation of the regulatory protein, releasing the hexokinase 4. If glucose concentration in the blood is high, then this is what happens. So we have the capillaries, that's glucose in your blood. It goes in through your GLUT2 receptor, and then it goes into, it's turned into glucose 6-phosphate, fructose 6-phosphate, and then it goes here. And then this is where you have your hexokinase 4. And then it can be taken into the nucleus. So increased transport of glucose into hepatocytes, that's just your liver cells. Hepato means liver via GLUT2. There's increased activation of hexokinase 4. It doesn't get saturated because it has a higher Km value, and it is released from the nucleus. So storage is extra glucose as glycogen. So this hexokinase 4 helps with that because you have so much glucose, you want to store it as glycogen so that you can use it later when you need it. Now, if the glucose concentration in the blood is low, then you decrease your transport of glucose into the hepatocytes. So this GLUT2 receptor would just go into the cytosol, and it wouldn't be here at the plasma membrane, because if you don't have a lot of glucose, you don't want to take it in. That's how you regulate how much glucose is in the blood. Second regulation point is phosphofructokinase 1 and fructose 1,6-bisphosphatase. So this is the next reaction that is irreversible. So in green, we have GO glycolysis if AMP concentration is high and ATP is low. And then GO gluconeogenesis if AMP is low. So I do think they're going to ask you about specific molecules and like whether they inhibit or promote glycolysis or gluconeogenesis. So I'd make a flashcard. Regulation of phosphofructokinase 1. So that's that really highly regulated enzyme for reaction number 3. So ATP is a substrate and also an allosteric modulator of phosphofructokinase 1. So we talked about this in discussion, how phosphofructokinase 1 has different binding sites for ATP. And depending on which binding site is being used by ATP, that is the effect that it will have on phosphofructokinase 1. So at low ATP concentration, the Km for fructose 6-phosphate is relatively low, enabling the enzyme to function at high rate at relatively low fructose 6-phosphate. When the ATP concentration is high for fructose 6-phosphate is greatly increased. The Km is greatly increased. So we do not want to spend glucose on glycolysis. So remember, when the Km is increased, it moves to the right, and that means that it's less effective. So that's kind of how it gets modulated. Either it'll increase the Km to make it more effective, or it will decrease the Km to make it less effective. Regulation by fructose 2,6-bisphosphate. Fructase 2,6-bisphosphate is not a glycolytic intermediate, only a regulator. Okay, so I think they might ask you about this one. So this enzyme, fructose 2,6-bisphosphate, that is not like a part of, or this molecule. It's not a part of our pathway. You know, the 10 steps in glycolysis, we have fructose 1,6-bisphosphate. But here they're talking about fructose 2,6-bisphosphate. So this is not a glycolytic intermediate. This means that it is not one of the substrates or products in our 10 steps. It's actually a regulator. So fructose 2,6-bisphosphate is produced specifically to regulate glycolysis and gluconeogenesis. So they might ask you about this. It activates phosphofructokinase for glycolysis, and it inhibits fructose 1,6-bisphosphatase for gluconeogenesis. So it activates this enzyme and it inhibits this enzyme. Okay, and that's fructose 2,6-bisphosphate. So it's used for regulation of the glycolysis and gluconeogenesis pathways. It promotes glycolysis and it inhibits gluconeogenesis. So insulin is when there's high glucose in the blood. Glucagon is when there's low glucose in the blood. And then what happens? What does it promote? So insulin promotes when you barely ate. So in the exam, I'm trying to tell you things the way I think she might ask them on the exam. So, because she wants you to go full circle with these concepts. So I think she might ask you, like, if you just had a cheeseburger, like, which of the following would happen? Well, you would get insulin, and that would increase the concentration of fructose 2,6-bisphosphate, and that would promote the process of glycolysis to make lots of ATP. Okay, so note, when the insulin concentration is high, you will make a lot of fructose 2,6-bisphosphate to stimulate glycolysis. And then when you're starving, like you've been in lab for 12 hours, you have low glucose in the blood. So glucagon will decrease fructose 2,6-bisphosphate concentration, and gluconeogenesis will be used. So this is where you use ATP to make new glucose. So glycolysis and gluconeogenesis are differentially regulated by fructose 2,6-bisphosphate. So remember, fructose 2,6-bisphosphate is not a glycolytic product. It's just used for regulation, and it promotes glycolysis and inhibits gluconeogenesis. Yeah, so go glycolysis if fructose 2,6-bisphosphate is high, and go gluconeogenesis if fructose 2,6-bisphosphate is low. Regulation of fructose 2,6-bisphosphate levels. Phosphofructokinase 2 and FBPase2 separate enzymatic activities of a single bifunctional protein. They are reciprocally regulated by insulin and glucagon through the phosphorylation. So insulin and glucagon, definitely make a flashcard for this down here. How if you have high blood glucose, you have high insulin, which will increase fructose 2,6-bisphosphate and increase glycolysis. And glucagon will stimulate gluconeogenesis. Definitely you have to know that. And then here we have, so these enzymes are reciprocally regulated by insulin and glucagon. Okay, so here we have insulin. I was hoping my pen would work, but nope, it's just, at least my computer. Okay. So here we have insulin, and here we have glucagon. So if you have a lot of fructose 2,6-bisphosphate, this stimulates glycolysis and it inhibits gluconeogenesis. So when you have insulin, you have water that breaks that phosphate, so phosphoprotein phosphatase. So you just have that alcohol group there. And then when you have glucagon, that increases cyclic AMP. And so this ATP is used and you phosphorylate. So we used to have the alcohol, but now you phosphorylate. So this is when there's a low blood sugar. You have glucagon, which increases cyclic AMP, and an ATP is used for phosphorylation. And this is a cyclic AMP dependent protein kinase. And this will inhibit glycolysis and it stimulates gluconeogenesis. Glycolysis and gluconeogenesis are differentially regulated by citrate. So here we go. Citrate inhibits glycolysis, but it promotes gluconeogenesis. So citrate can be made from other sources like fatty acids and amino acids. It will inhibit glycolysis and promote gluconeogenesis. So just in case you forgot, citrate is a part of the citric acid cycle, and it's when oxaloacetate, that 4-carbon molecule, is combined with the acetyl group from our pyruvate, and we end up with our 6-carbon citrate. So again, it inhibits glycolysis and it promotes gluconeogenesis. So citrate is a key intermediate in the aerobic oxidation of pyruvate, fatty acids, and amino acids. So stop glycolysis if citrate is high. Yep, we talked about that. Citrate inhibits glycolysis. So high citrate concentration increases the inhibitory effect of ATP, further reducing glycolysis. It is a signal that enough energy is being obtained from fats and proteins. Third regulation point is pyruvate kinase. So here we talk about what happens only in the liver and all glycolytic tissues, including the liver. So I'd make a flashcard here. So in the liver, inactivated by phosphorylation in response to signs of glucose depletion, glucagon. Glucose from the liver is exported to the brain and other vital organs. So if your blood glucose is low, we know that your brain like really needs glucose. So in the liver, glucagon will stimulate phospho or pKa. It'll use ATP to phosphorylate pyruvate kinase L, which is inactive. And then so that the whole goal is that the liver won't be using glucose. Instead, it'll be like exported to your capillaries so that that glucose can be used by your brain and the other organs. So here there are at least 3 isozymes in all the other tissues, allosterically activated by fructose 1,6-bisphosphate. It increases the flow through glycolysis, allosterically inhibited by signs of abundant energy, supply all tissues, ATP, acetyl-CoA, and long fatty acid chains. Alternative fates of pyruvate. Okay, so we talked about that earlier. Pyruvate can either go down to the citric acid cycle, or it can be used for, fermentation. So pyruvate can be a source of new glucose. Oh yeah, you can also go backwards and make glucose. And you can store energy as glycogen. Pyruvate can be a source of acetyl coenzyme A. Yeah, you can go into the citric acid cycle. You can store energy as body fat, or you can make ATP via the citric acid cycle. Acetyl coenzyme A stimulates glucose synthesis via gluconeogenesis by activating pyruvate carboxylase. So yeah, it just depends on which enzyme is being activated. The amount of many metabolic enzymes is controlled by transcription. Insulin promotes glycolysis. So stimulates the transcription of genes that encode hexokinase 2 and phosphofructokinase 1. Yeah, so if insulin promotes the process of glycolysis, so we had 10 reactions and 10 enzymes, so it would make sense that insulin would also stimulate the production of more of those enzymes. And glucagon is the opposite. It promotes gluconeogenesis. So it makes sense that it would stimulate the transcription of those genes for gluconeogenesis, like um, the four enzymes that are required to bypass those three irreversible reactions from glycolysis. Okay, does anybody have any questions? We're moving on to lecture 25. Okay, control glycogen. Oh, 25. This one's short, thank goodness. Okay, metabolism of glycogen in animals. So glycogen is that really where we stored a bunch of the glucose monomers. Excess glucose can be stored as glycogen. Glycogen is kept in large cytoplasmic granules. Glycogen storage occurs mainly in the liver. It's the reservoir for other tissues, especially the brain. So anytime our blood glucose is low, the liver can break down glycogen and throw in a bunch of those glucose into our blood so they can reach our brain. So in the brain, glucose lasts 12 to 24 hours. And in the muscle, glucose only lasts about 1 hour. So glycogenolysis is when glycogen is degraded to glucose units for the use of energy production. So glycogenolysis. Remember, lysis means breakdown. And then genesis means to make something brand new. So glycogen can be made from excess blood glucose. Like I love boba. I drink boba like 3 times a week. So anytime I do, my body definitely has to undergo this process of glycogenesis because there'll be too much glucose in my blood. So it'll have to go in and make glycogen or recycling of glucogenic metabolites like lactate or certain amino acids. So here's the mitochondria in our liver cells. And then these dark granules are glycogen. So if you can imagine just like thousands of glucose monomers attached to each other and clumped up, and that's what glycogen is. And it's just waiting there to be used. Glycogenin starts a new glycogen chain. It's like glycogenin. New glycogen particles begin with the autocatalytic formation of a glycosidic bond. Glycosidic bond is just a bond between two sugars. The glucose of UDP glucose and tyrosine residue of the protein glycogenin. So, structure of the glycogenin particle. Each chain has 12 to 14 glucose residues. So this is glycogenin. In red, we have a primer. In purple, we have the second tier, third tier. Green is the fourth tier, and blue is the 5. So 55,000 glucose residues, 55,000 glucose residues in one glycogen. So up here we have UDP glucose, we have glycogenin, which is that protein that happens to have a tyrosine amino acid with an alcohol group. And so it binds UDP glucose. And then we have a chain extending activity. So when this primer is long enough, extension continues with glycogen synthase. So this enzyme, glycogenin, it just gets the process started. And it's 12 to 14 residues, and it's considered a primer. So glycogenin makes the primer. And after that, you have glycogen synthase enzyme that will go ahead and continue adding more glucose to that glycogen molecule. So glycogen is synthesized by glycogen synthase. Yeah, that's the enzyme. And so UDP glucose, sugar nucleotide. Glycogen synthase transfers the glucose residues of UDP glucose to the non-reducing end of a glycogen branch. So they might ask you about that, like to which end, the reducing end or to the non-reducing end, and it's to the non-reducing end. Synthesis of branches in glycogen. So there's a non-reducing end. Glycogen core, glycogen branching enzyme. So yeah, you have another enzyme that's for branching. So yeah, these are all the non-reducing ends. And these are alpha 1,4 linkages. And for the branches, it's alpha 1,6. So they might ask you a question on the final about the connections. So alpha 1,4 is for linear and alpha 1,6 is for branching. The glycogen branching enzyme, amylo 1,4 to amylo 1,6 transglycosylase or glycosyl 4,6 forms a new branch point during glycogen synthesis. Glycogen and starch. So same thing, glycogen is in us and starch is in plants. When needed, units are removed one by one from the non-reducing end. So they might ask you when you need to remove one of the glucose monomers from glycogen, where does it go from? And you take it from the non-reducing end. So that's these right here. So glucose residues are removed by glycogen phosphorylases. So yeah, glycogen phosphorylase is the enzyme that removes the glucose monomers from glycogen. Phosphoglucomutase interconverts glucose 1-phosphate and glucose 6-phosphate. So that's pretty cool. This enzyme can just go in and transfer the phosphate from glucose 6 to carbon 1, carbon 6 to carbon 1, or vice versa. In glycogen degradation, glucose 1-phosphate is converted to glucose 6-phosphate for glycolysis. Yeah, because for glycolysis, we need glucose 6-phosphate first. So in glycogen synthesis, XX glucose 6-phosphate is converted to glucose 1-phosphate. So that makes sense for glycogen synthesis, we want glucose 1-phosphate, but for glycolysis, we need glucose 6-phosphate. Dealing with branch points in glycogen. Glycogen phosphorylase works on non-reducing ends until it reaches 4 residues from an alpha 1 to 6 branch point. Debranching enzyme transfers a block of 3 residues to the non-reducing end of the chain. Debranching enzyme, so there's another debranching enzyme, cleaves the single remains alpha 1 through 6 linked to glucose, which becomes a free glucose unit. Bifunctional debranching enzyme alpha 1,6 through alpha 1,4 to glucotransferase. So here's the non-reducing end, and this is the alpha 1 to 6 linkage. That's how you know it's a branch. And then you have this enzyme glycogen phosphorylase, which breaks glucose 1-phosphate molecules. And then you have transferase activity of debranching enzyme. You basically just moved those residues, and then you have glucose. Glucose 6-phosphate is dephosphorylated in the liver for transport out of the liver. So glucose 6-phosphate is the one we use for glycolysis. And you have to remove that phosphate in the liver so you can get it out. So glucose 6-phosphatase is an integral membrane protein in the endoplasmic reticulum. So phosphatase removes a phosphate. Glucose 6-phosphate transporter 1 carries the substrate from the cytosol to the lumen. Glucose and phosphate pass to the cytosol on specific transporters, transporter 2 and 3. And glucose leaves the cell via the GLUT2 transporter in the plasma membrane. So glucose can't be phosphorylated if it's going to leave the cell. As long as glucose is phosphorylated, it is trapped inside of the cell. So here they're talking about how they get rid of the phosphate and then how glucose leaves through its transporter 2 into the capillary to increase blood glucose concentration, like we said, for the brain and for the other organs. Integration of glycogen synthesis and degradation. Okay, so this is something that I would definitely make a flashcard of right here. From glucose, you can use hexokinase to make glucose 6-phosphate. So hexokinase will add a phosphate to carbon number 6 and glucose. And then from there, you would use phosphoglucomutase to convert carbon 6 to carbon 1 if you needed to transfer the phosphate from carbon 6 to carbon 1, depending on what pathway you want to go to. So glycogen phosphorylase would take away that phosphate that you had placed on carbon 1 to make glycogen. Or actually, no, it wouldn't take away the carbon. It would go into the glycogen chain. So here, if you have glucose 6-phosphate and you want to remove that phosphate, you use glucose 6-phosphatase. The phosphatases are the ones that remove the phosphates. From glucose 1-phosphate, if you want to go to glucose 6-phosphate, you just use the mutase, phosphoglucose mutase. Now, if you want to go to UDP glucose from glucose 1-phosphate, you would use UDP glucose pyrophosphorylase. So here it's kind of like about learning the names of the enzymes and what they do. So glycogen synthase would take you from UDP glucose to glycogen. And glycogen phosphorylase would take you from glycogen to glucose 1-phosphate. So I would learn these enzymes and what they do, what is the substrate versus product, and then think about like when they would be used. So if you want to break down glycogen and go to glucose 1-phosphate, then you would use this enzyme glycogen phosphorylase. So when would that happen? So this would happen if you haven't eaten in a really long time, right? You need to break down that glycogen, or if you've been working out like way too much. Right, so you want to break down that glycogen to make more glucose to get more energy. So like glycolysis and gluconeogenesis, regulation occurs at irreversible points in the pathway. Control of glycogen synthesis and breakdown. Glucagon indicates low blood glucose, and epinephrine indicates the need to fight or flee. So if you have a lot of ATP, that promotes cyclic AMP, and then protein kinase A will promote protein kinase A, phosphorylase kinase, phosphorylase. I don't think she's gonna ask anything about like too specific about this. But basically everything green is more active when you have a lot of ATP. Control of glycogen synthesis and breakdown. Okay. So epinephrine from vigorous muscle activity. So when you're working out a lot, and glucagon in the liver, they trigger the phosphorylation of phosphorylase B. So phosphorylase B is glycogen is not degraded. So when you have epinephrine and glucagon in the liver, this triggers phosphorylation of phosphorylase B, converting it to phosphorylase A. With phosphorylase A, that's the active form, and this means that glycogen will be degraded. And that makes sense because if you're working out a lot, then you're going to want to degrade glycogen so that you can get glucose, so that you can go through glycolysis and make more ATP. So that's what Maria wants you to be able to get at. So yeah, glycogen degradation would increase. So if you have epinephrine and if you have glucagon in the liver, this will trigger phosphorylation of phosphorylase B, converting it to phosphorylase A, which is the active form, which will increase glycogen degradation. Now, if you have PP1, phosphoprotein phosphatase 1, remember, phosphatases remove phosphoryl groups. So this will remove the phosphoryl group from group A, and then this will form phosphorylase B, this guy right here. So glycogen degradation would decrease. So yeah, there's different conditions, but anytime you remove the phosphate here, it will give you the less active form. You won't have glycogen degradation. So that means that you don't really need to make ATP. You already have ATP. And now insulin. So insulin up here is when you have high blood glucose. So you just ate a cheeseburger. Glycogen will be synthesized because you've got too much glucose in the blood. And yeah. So insulin inactivates GSK3. So that's why you have insulin with an X for GSK3. It inactivates GSK3 and it activates phosphofructokinase 1. Where am I? Yeah, PP1, which is glycogen synthesis increases. So insulin inactivates GSK3 and it activates PP1 so that you can make glycogen. And that makes sense because you just ate like a really big meal and you've got too much glucose in your blood. So you want to store that glucose safely. So you want to make glycogen. So the way that insulin does that is that it inactivates GSK3, but at the same time, it activates PP1. So glucose 6 phosphate acts allosterically to make glycogen synthase B, a better substrate for PP1. Okay, so here we have PP1. Phospho, I've seen a question on an exam about PP1. So phosphofructokinase 1, insulin activates it, glucose 6 phosphate activates it, glucagon and epinephrine inhibit it. So whenever PP1 is activated, you're going to make an glycogen to store the glucose monomers. But whenever you have glucagon and epinephrine, that will inhibit PP1. So you will not be making glycogen. Control of carbohydrate metabolism in the liver. So what happens when you have high blood glucose? Your insulin level increases. So your insulin sensitive protein kinase, you increase PKB, increase synthesis of hexokinase 2, phosphofructokinase 1, and pyruvate kinase. You also increase your GLUT2 receptors in the plasma membrane. So you can increase the amount of glucose that you take into the cells because you want to get it out of your capillaries. And then this will lead to glycolysis. And then over here, the synthesis of these molecules, these are the transcription regulation. So you're increasing the transcription of all the enzymes that you're going to need for glycolysis. And then increase PKB, decrease GSK3, increase glycogen synthase. So you have a lot of glucose in the blood, so you're going to go ahead and make that glycogen. So when you have a lot of insulin, again, you're going to go ahead and increase your insulin sensitive protein kinase, increase PP1, which also helps you decrease glycogen phosphorylase, and you decrease the glycogen breakdown. So the key here is knowing your enzymes and knowing what function your enzymes have. Once you know that for both gluconeogenesis and glycolysis, you will be able to answer all of these questions, no matter how tricky or hard or complex they are. So high blood sugar leads to increased glycolysis and glycogen synthesis and decreased glycogen breakdown. So that makes sense. If you're making glycogen, you can't be breaking down glycogen too. So you have to inhibit those enzymes that break down glycogen, and you got to promote the enzymes that promote glycogen synthesis and glycolysis. Okay, give me a second. Okay, I definitely make flashcards for this, for high blood sugar and low blood sugar. So low blood sugar would decrease glycolysis. That makes sure you don't want to break you don't want to break down glucose for ATP if you don't have glucose, and it decreases glycogen synthesis because you don't have enough glucose. So how are you going to make glycogen? But it increases glycogen breakdown. So yeah, if you have low blood sugar, you're not going to make glycogen. Instead, you're going to tear off the individual glucose molecules so you can put them into the blood for the brain and stuff. So yeah, definitely what I would do is make, like, maybe on a piece of poster board, like write these out and then go ahead and write down like the definitions for each of these, because then it'll help you make sense of everything and you'll be ready for these difficult questions. But this is probably like the most difficult slide because it's combining everything now. Control of glycogen synthesis by insulin. Insulin increases the uptake and phosphorylation of glucose in muscle, and it stimulates glycogen synthase. So insulin indicates high blood glucose. So this is what we covered earlier, how you have the receptors inside of these vesicles, but whenever you have insulin and insulin attaches to the receptor, all of a sudden all these vesicles go ahead and they merge with the plasma membrane and they bring in all the GLUT2 or GLUT4 receptors so that they can take all of the glucose from your blood so that you don't have too much blood glucose. Okay, and here I wrote down some control of carbohydrate metabolism. So glucagon is when you have low energy and we need to make more ATP. So it happens in the liver, and in the liver, glucagon stimulates the degradation of glycogen so that individual glucose molecules can be released. So this is what I mean when I tell you to go ahead and write down the definitions or like notes of things so they can help you learn. So again, glucagon in the liver, you have glycogen. Glycogen is broken down to glucose 6 phosphate, and glucose 6 phosphate will be dephosphorylated in the liver, and then it will be sent as glucose to the bloodstream. You can also have pyruvate in the liver. Glucagon also stimulates gluconeogenesis from pyruvate, and it'll send it to the blood. So yeah, if your blood glucose is low, your body will do just about anything to make sure that it can give you more glucose. So you have pyruvate here that is being turned into glucose and then sent to the blood. And then glycogen is being broken down into glucose 6 phosphate, and then that is dephosphorylated before it's transferred to your blood. So then over here in the muscle, you can break down glycogen. So glycogen is broken down into glucose 6 phosphate, and here instead of transferring it to the blood, you want to use it. So here the goal is to use that energy. So you go down to pyruvate and then all that. And then glyconeogenolysis. This is the breakdown of glycogen to get individual glucose molecules. So that's how it happens in the liver. That's how it happens in the muscle. And remember, the goals are different. In the muscle, you want to use the energy immediately, but in the liver, the goal is to break everything down or to create new glucose and send it to the blood so that your brain and everything else can have enough glucose. Okay, so this is kind of important. Cascade mechanism of action. So this is a myocyte, which is a muscle cell, and this is a hepatocyte, which is a liver cell. And you have epinephrine and you have glucagon. And so here you're going to break down and get 20 times the amount of molecules with just one signal. And then this will just increase and increase 100 times the amount of molecules, 1,000 times the amount of molecules. So the binding of one hormone to its receptor can lead to the release of 10,000 molecules of glucose that can be released from glycogen so that it can go to the blood from the liver or in myocytes for glycolysis. This is an amplified process. So that's really interesting. So here, like, remember that one glycogen has about 50,000 glucose monomers. And when one hormone binding, like one hormone binds, you can release 10,000 of those glucose monomers from glycogen. And if you're in the muscle, your body will use it right away. But if you're in the liver, then those 10,000 glucose monomers will go ahead and be dephosphorylated and then sent to the blood. So that's pretty crazy. It's an amplified process. Okay, so that's the last thing that I'm going to go over. Does anybody have any questions? Since our time is up. The good thing is that 26, this, I covered it in detail in our lecture, in our last discussion. So I went over the whole citric acid cycle. And then 27, Maria did a beautiful job of explaining this one. She already uploaded the video, so. No questions? Okay.